All right, it's my pleasure to introduce our keynote speaker for today, Dr. Mark Fast. He's a professor of fish health and comparative immunology at the Atlantic Veterinary College at the University of Prince Edward Island in Canada. His research group investigates how the environment impacts host pathogen relationships using genomic techniques, in particular with respect to how this affects cultured species such as salmon. For over 20 years, Dr. Fast's research group is focused on the host-parasite relationship of sea lice and salmon, such as the CRISPR-resist, genolice, and lice-resist. And since his postdoctoral fellowship at the Institute for Marine Bioscience, he's also been involved in vaccine development, patenting a few vaccines for salmon, and most recently investigating the application of mRNA vaccine technology to Salmonella diseases. He's seen a lot, done a lot, and we're looking forward to the presentation, Mark. Thanks for being here.
Great. Thank you very much, Nick. And thanks for the opportunity. You hear me okay, Nick? Just want to make sure.
Yep, sure can.
Excellent. Okay. Definitely not the best for last, but I appreciate the sentiment. I wanted to kind of go over a broad kind of overview of salmon aquaculture and how there's been essentially major changes to the industry over the last decade or more, and how that's altered both the industry's kind of disease focus, but then also, research labs focus as we are interested in host pathogen interactions, but also applied research in aquaculture. So in salmon aquaculture, probably the greatest cost in money and effort is in terms of disease is sea lice, and that tends to work out pretty well for myself and my research group, since that's one of our major research focuses, or foci, I guess. I've talked about this before here and in other places. I'm sure many of you are familiar with sea lice, so I won't go too much into the general or the background in that. We're really dealing with two main species, Leptopteris salmonis and Caligus rogercressii. And Lep salmonis impacts the northern hemisphere, both in the North Atlantic and North Pacific. I see Lars Hamre on there. He's published many things. He's a great mind in terms of Leptopteris salmonis and sea lice in general. We've worked with Lars and a group in Norway, and then also with Caligus rogercressii in Chile for salmon aquaculture down there. And really the biggest issue with the main issue with sea lice is we get spillover infections generally in the spring. These increase through the fall with little recruitment in the winter. And there's a major concern for spillback onto wild populations. And you can see by this graph of kind of treatment usage in Norway from 2000 to 2016, that pretty much as salmon are being produced, around 2008, 2009, the amount of salmon being treated specifically for sea lice began to really take off. And that was really around a lack of efficacy of the drugs that were being used for sea lice. And it really kind of made me think of the title for this talk. It really changed kind of the landscape or the aquascape, pardon my dad pun, for the industry in terms of how to control lice. And then you can see that that red line drops way off after 2015 into 2016, and that's been maintained for the last decade, where there's been a shift to other treatments or other intervention strategies for sea lice. So there's a great paper that came out from Carrie Helgeson and her group a couple of years, I think it was 2 years ago, and I wanted to use it here to kind of use as a backdrop for this talk. And this looks at kind of a 19-month seawater grow out in Norway, two particular farms, and just kind of showing lice counts and some of the intervention strategies that are used in these at these farms, and how that's changed quite a bit. So if you look over the 19 months in kind of the top farm and the bottom farm, you can see that in these cases, the fish are being stocked in the fall here. And shortly after the fish are going in, there's some medicinal treatment. In the top farm, amamectin benzoate. On the bottom farm, teflubenzuron. These are an in-feed treatment that takes anywhere from 1 to 2 weeks, depending on the dosage, to control lice infection. And then you have a series of other interventions. The blue lines in the top one showing freshwater treatment, thermal treatments. The bottom graph showing thermal, mechanical, different treatments. There's also lumpfish and wrasse being stocked, and quite a few treatments occurring over the 19-month period. Now, some of the newer I guess newer treatments that have come online over this last decade are these freshwater thermal and mechanical treatments. There is also peroxide, but that's been around for a while. You'll also notice that, between July and September, temperature's quite high, so it's above, or at least for us northerners, us salmon people, above 15 C, and that's there's half a dozen treatments that are occurring in there. That's because the lice themselves are their generation time reduces drastically as temperature increases. So you have more recruitment, greater infection level, and so higher treatments required. So as I talked about kind of before, if we look at drug application and why kind of the industry's gone away from some of these drugs, in that top farm, amamectin benzoate was used kind of at the beginning of the production cycle and not again, and that's because of the resistance issues around amamectin benzoate. And what's kind of interesting is that regardless of the species or even subspecies of lice that we look at, we get the same chromosome associated with resistance. So if we look at Kevin Glover's work on the Atlantic Lepsalmonas and Amber Mesmer and our work on the Pacific subspecies of Lepsalmonas, we can see on the right we have the chromosomes kind of lined up and individual single nucleotide polymorphisms that are associated with resistance in these two populations occurring on chromosome 5. So the top graph is actually, or the top figures are from Kevin Glover's group's work, and you can see that's the 5th chromosome, and the bottom graph is from Amber Mesmer's work. Now, despite these SNP single nucleotide polymorphism chip papers that have come out and an understanding that there's a lot of polymorphisms on this particular chromosome, it hasn't really resulted in any working working diagnostic tests that can kind of inform us when a population is tolerant or resistant to this drug. The graph the picture on the far left shows kind of what we still the industry still uses in many places, and that's bioassays to confirm whether a population is tolerant or sensitive to the drug. Now, modeling, some of the modeling work that our group showed with Gregor here pictured is that there was lower risk for areas in terms of resistance development, whether it was monogenic or polygenic, like we think amamectin is, for areas with healthy, large wild populations due to the likely refugia. So as long as there's a decent number of wild salmon that have lice that aren't getting treated with any particular drug, the likelihood of resistance development, it will be slowed. And if there's a fitness cost to this resistance development, it will slow it even further. However, if we look at in BC, where we've done some work, and there's very large wild populations of Pacific salmon. That refugia idea seems to have worked in the past. However, more recently, there's a study that came out in 2022 looking at efficacy of amamectin benzoate, and starting to see, again, looking at these, using these bioassays as an indicator, you can see that in the last couple of years, the EC50s for the bioassays have been trending upwards. And if you look back more kind of 2014, 2015, 2016. That's in the time frame where we published the work with Amber Mesmer on the mechanism of resistance, or this, polymorphisms associated with resistance in BC. So as Eminem says, back to the lab again, right? So there's still a need for diagnostic tools for emamectin. It's still used in the North Atlantic, still used in the North Pacific. So we've actually had our student here, Shelby Reimer, working not just on using a single nucleotide polymorphism chips, but actually looking at whole genome sequencing of wild and cultured, or salmon from wild and cultured fish, to get an idea of honing in better on what the mechanism of resistance might be, and come up with a better diagnostic tool for this resistance. And the difference here being that in Shelby's work, she's using bioassays as a differentiation tool. So kind of bringing the two together, both sequencing of tolerant and susceptible populations, but separating those out based on the bioassay, and then looking at SNPs associated with resistance. One of the things that Shelby has kind of shown in her preliminary work is that SNP distribution is already quite high in chromosome 5, regardless of whether the lice are tolerant or susceptible. And so if you think about how a chip is, where some of these other publications, including ours, have come from in the past, is you're actually looking at interesting areas within the genome, but also spacing out individual SNPs as they occur throughout the genome on the chip, so that you can have the best coverage of potential polymorphisms associated with resistance. However, there will be a bias for those chromosomes or those areas that have more SNPs in them. So that probably contributes to us finding chromosome 5 as an important chromosome. So we're kind of getting back to looking at that data currently, and trying to assess or come up with a better diagnostic tool for emamectin. What was that other drug that I mentioned there? Teflubenzuron. So in the bottom graph, teflubenzuron has been used recently as well. It belongs to the benzoylurea class of compounds, and I've talked about these a little bit before. And you've probably seen presentations from our former students, Jordan or Dylan, as they kind of worked through both the molting or chitin synthesis pathway for Lepsalmonus and for Caligus rhodochrosii, because these are highly conserved pathways throughout the arthropoda. And the benzoylureas really act as chitin synthesis inhibitors in arthropods in general. And they kind of confirmed some of that work. So teflubenzuron being one of these BPUs. We've done more work on lefenuron and some of the other BPUs, but we've had some interesting findings about teflubenzuron. In this case, I just wanted to highlight, if we look at the chitin synthesis pathway, starting from glucose and kind of making our way down to chitin synthase and actually production of chitin, we can see that there's this particular gene which codes for the phosphoacetylglucosamine mutase. Try saying that 3 times fast. And it actually has a positive correlation with lefenuron in terms of dosage. So as you increase lefenuron dose, you actually see this gene get upregulated in Lepsalmonus, and we actually see that in Caligus rhodochrosii as well. So if we look here, this just shows that let me go back and remember the name. Phosphoacetylglucosamine mutase is also referred to in short, so you don't have to keep saying that, NST in Drosophila and other arthropods. What we actually see, if we move a little further up the chain, the chitin synthesis pathway, GFAT is a gene that has opposing regulation to NST. So as NST goes up, GFAT goes down, just as Homer's showing in the picture to the side. What's interesting is that, again, if we look at lefenuron or we look at hexafluomeron, a couple of these compounds, when GFAT, when they're exposed to, when lice are exposed to any of these BPUs, GFAT actually is induced. So we actually can show that not only does GFAT have opposing regulation to NST, but it has opposing regulation to it when associated with BPU treatment. And this is actually in the middle picture there showing as the copepodid stage ages, and they are this is Caligus Roger-Cressii data, are exposed to lofenuron. We actually can see that GFAT expression is downregulated by lofenuron treatment. So where this kind of comes together is if we look at them as a group, teflubenzuron, hexaflumaron, lofenuron, and we examine their impacts at different dosages to copepodid settlement and development on salmon, we can actually see that some of these other compounds, hexaflumaron, lofenuron, do a little bit better in terms of protection, in terms of at the same dosage as teflubenzuron does. But one of the things we also see is that there's a shared suite of genes across these compounds in terms of how the louse responds to them. So there's shared genes between the response to lofenuron and hexaflumaron. And if we look at teflubenzuron in kind of that salmon colored, most of the genes that are differentiated or differently expressed in lice when they're exposed to it are shared with lofenuron or hexaflumaron. So these are all part of the same class, and the potential for them to induce similar responses in the louse is quite high. Now, teflubenzuron is utilized in Norway. Hexaflumaron and lofenuron are approved for use in Chile. So the risk of utilizing multiple BPUs is low, but there is hexaflumaron and lofenuron used in Norway, as I mentioned. One of the interesting parts of the teflubenzuron responses is that we do see induction of not just the molting genes or molting impacts by teflubenzuron, but induction of some of the multi-drug resistant proteins or multi-drug resistant genes in lice that survive. Now, we see some of these impacted by EMB, but also multiple other drugs. So there's a risk there in terms of its usage, and not surprisingly, probably with other compounds, how that might interact with drug usage. Now, again, getting back to that graph, you can see that for the most part, medicinal treatments are used very sparingly and usually at the early stages to kind of provide a lengthy protection before lice initially settle. These molting inhibitors especially aren't going to impact adult stage lice because they don't molt anymore, but can actually provide protection due to settlement or initial molting. So if we can't control lice with medicinal treatments, what about freshwater, thermal, mechanical delousing? And this is kind of where the industry has moved towards, as I said, over the last decade. It involves fish being pumped out of the cage and then going through either something like the hydrolyser or thermal lyser. Hydrolyser being high pressure water that is, I guess, shot on across the fish to dislodge the lice. Thermal lyser, where the fish are exposed and the lice are exposed to slightly or increased temperature. There's, of course, welfare concerns from the resulting gill damage, as well as concerns about removing the mucus layer. Matt, I think, is still here. We just got an email regarding this just moments ago. It's a major issue for concern for salmon welfare and salmon health. And then you also have the combined impacts potentially of efficacy at high temperatures. So if you're increasing the temperature of the water to dislodge the louse, what happens in these cases where water temperatures are increasing already and in the middle of the summer? So that's where we've had this shift from infectious disease control to actually examining control of or trying understanding the unintended impacts of some of these treatments. So we've actually shifted gears in some cases to looking at kind of gill health and potential complex gill disease, other things that are impacting the health of these salmon that might be induced due to gill trauma or other trauma that occurs on the farm. So in particular, this gill health initiative that we've been working on this last couple of years, we're funded by Genome Canada and Genome BC and sponsored by industry, Grieg and Cermak, as well as Cargill, or to try and look at lab models of gill trauma, whether it's hydrogen peroxide treatment, freshwater treatments, as we've talked about, even jellyfish exposures, because jellyfish exposures have increased, or jellyfish blooms have increased with increasing temperature over time, and seeing how we can identify sort of sensitive markers to describe the traumatic events and describe early stages of gill disease in these fish and how we might resolve it. So not exactly looking at the infectious disease sea lice or even the potential of other infectious diseases that cause gill trauma in a lot of cases, but looking at developing models for gill trauma in the lab so we can come up with better solutions in the field. So, as I mentioned, this is involved. We've had a student working on looking at gill damage in the due to nematocysts or gill damage due to jellyfish. If you would have told me, 10 years ago, we'd be working on jellyfish impacts on the gills, I would have laughed. But it's a real thing that's impacting gill health. These nematocysts damage the gills. They release toxins once the nematocyst comes in contact with the host cell, but also toxins are released into the water column once these nematocysts evert and release due to changes in pressure. So we've developed a in vitro model looking at jellyfish toxins. This just as an example of ASK cells and cytopathic effects of jellyfish toxins. We've looked at identifying or generating individual markers for both acute and chronic impacts to the gill and to these cell culture in vitro kind of proxies. And that's work that was done both by our student Crystal Veltman and Albert Caballero Solares, and then look at application to the whole animal. So exposure of jellyfish to the gills of these fish, and how does that impact general gill health and overall fish health in this system, and then trying to look at both histologically what the impact is. So we have our friend Dave Groman who goes through and scores the histological challenges or changes that occur in the gill, and we're trying to then assess how those correlate or don't with more sensitive molecular markers following the same treatment. So jellyfish, for instance, causes quite an acute impact. We see when we look at our PCAs that the molecular markers correlate well with the histological descriptors, but in several cases are quite a bit more sensitive. And then if we look at other measures of gill trauma, like a chronic gill trauma, such as freshwater bass, or in this case, this is looking at hydrogen peroxide bass, repeating the exposure 3 to 4 times every week, we can see again that some of these same markers are actually increasing in the gill, both not just at acute, but also chronic treatment with hydrogen peroxide, and that they're also impacted based on dose of hydrogen peroxide and temperature that they're exposed to. So these experiments, different ways of kind of initiating trauma in the gill, have allowed us to get to a point where we have a few individual markers of both acute and chronic impacts on the gill that are useful in describing or at least showing us impacts on the gill prior to histological damage in these fish. So now the goal is to kind of look at some of these individual markers at the field level and see whether they can be predictive or useful, associated with some of the intervention strategies that occur on the farm, whether they're hydrogen peroxide, whether it has to do with a Cattosaurus or a jellyfish bloom, or any of the other environmental issues that occur in these farms. Another component of that work, and I don't have the time to really go into it, is Faru and Albert have done quite a bit of work looking at gill regeneration, especially Faru, and how removal of components of the filaments, or the distal ends of the filaments, how they can regrow, and what the impact is to these filaments as they're regenerating. How functional are they after regrowth, and what impacts the regrowth. So again, looking at this involves trimming the distal end of the filament, but we do see damage to, and in some cases complete removal of gill filaments under some of these hydrolyser or some intervention strategies that occur on the farm. So we're able to now start looking at individual markers of initial stages of gill regeneration, as well as kind of resolution of regeneration, and what occurs kind of 20 weeks out when in terms of the function of these gills once they fully regenerated. So that's some really interesting work that Faru's working on. But again, all coming out of developing a model to look at these unintended impacts that might occur on the farm due to intervention to control lice or other diseases. So as I mentioned, we have talked about kind of the impacts that we can have due to issues that involve medicinal treatment, some of the potential issues that occur following freshwater, thermal, or mechanical treatments that occur. But as I mentioned, we also have the potential for higher temperatures occurring on the farm for longer periods of time with coastal climate change in the oceans and increasing coastal temperatures. We've done some prior work a couple years ago with Sean Godwin, just showing the physiological impacts on salmon as they go to higher temperatures, but also when they're infected with different infection levels of lice. This just shows survival curves comparing temperatures from 10 to 22 degrees and looking at 0, low, and high infestation levels of lice. Low being less than 1, high being, I think it was 8 or more in this study. I'd have to go back and double check. But essentially showing survival probability decreases as temperature increases as lice infection increases. So more recently, one of our postdocs, Reza, has been doing quite a bit of work looking at temperature effects, both on sea lice infection and how that interacts with family background. So looking at different families of salmon with differing natural resistance to sea lice, and also looking at infection and reinfection, whether once the fish have gone through a treatment, what the subsequent genetic background means in terms of susceptibility to reinfection. In this case, I'm just going to show a little bit of data from Reza's work where he's looked at 2 temperatures, 10 and 20, so kind of extreme temperatures, but temperatures that in the Bay of Fundy, Atlantic Canada, fish will come across in the midsummer for extended periods. And what is the impact at different life stages, the calamus or the adult life stage, in terms of the host's ability to respond. So there's quite a bit of work that Reza's done, a lot of sequencing, and really looking at both the local site of infection, so what's happening in the skin, and also what's happening systemically in the kidney. Now there are some shared responses regardless of family background, whether they're susceptible or less susceptible. I won't call them resistant. And these change based on temperature, whether the animals are 10 or 20. However, it does not appear that resistance or relative resistance, changes based in the family based on temperature. The lice do better at 20 degrees in terms of their the impacts on the host. The lice, I shouldn't say the lice do better, but the impacts of the lice are greater at 20 degrees on salmon than they are at 10 degrees. And we see that both in the kidney and in the skin. But we see a much greater degree of difference across the families at the skin level compared to systemically in the kidney. So there are quite a few differences between families, but these are more, there's a greater difference occurring at the infection site in the skin than occurs systemically. And that's probably not a surprise. However, we do see that the terms or the functions associated with rejection of lice are quite common. And if we look across species to say something like coho salmon, which truly is resistant to the lice, we see similar responses in terms of impacts on chemotaxis, the C-type lectin signaling and cell adhesion molecules, and the antigen presentation. These are all impacted and are all important in rejection of lice. It just appears that they are not as heavily induced in Atlantic salmon as they are in the species like coho as part of the rejection mechanism. And we see that across families, some of these same genes that are important or associated with healing in the skin, like the matrix metalloproteases, are inhibited at the higher temperature, but are also inhibited by the louse. So we can see that MMP9 here, as an example, is a great indicator of chronic wounding and the wound healing process in the skin. We see it inhibited due to the louse infection, but we also see that we look across different families across temperature, it's also down-regulated or expressed at a much lower rate at the higher temperature. So these things together work to combine to cause these negative impacts, regardless of family and regardless of their level of sensitivity in terms of infection level. Because we're really talking about when we say the sensitive and the less sensitive, we're talking differences of maybe 20 to 30% and maybe in the odd case 40 to 50% in terms of the lice infection level, but they're always, the prevalence is still the same. It's still 100% prevalence of infection. So our lab has spent, its focus is on host pathogen interactions, but as the industry has changed and altered some of its focus, so have we. We've started to look more at drug mode of action and resistance monitoring, as well as stress, trauma, and welfare associated with some of these treatments and how they can be impactful in terms of salmon health. And then moving into both breeding and genetic background in terms of their ability to show or make improvements in terms of resistance to sea lice and enhancing salmon health on the farm. A part of that too is genetic manipulation. So where do we go in the future? So obviously there's a debate and a discussion that's been going on for a while about genetically modified organisms, about gene editing to improve disease resistance. We have a pretty common view about the negativity around genetically modified organisms. But we also have the opportunity to look at gene editing to improve disease resistance, and gene editing, such as CRISPR-Cas9, provides non-foreign DNA insertions or edits. So it's not considered GMO, and therefore I think it has the opportunity to teach us a lot about the genetic controls for diseases such as sea lice, but also the potential for ways of controlling lice in the future. So that's kind of where some of our research is going now, is looking at knocking out and potentially knocking in function for particular genes in multiple species of salmon, and how that impacts their disease resistance, in particular to sea lice. But it can be used across multiple diseases and disease agents, and I think that's where, or particular pathogens. And I think that's where the industry and a lot of the research is gonna go. So here's just an example of our current work, looking at, these are juvenile unedited coho salmon and our albino coho salmon on the left, with a particular edit to knock out pigmentation. And what we would call the one on the right is our mosaic, because you don't always have 100% knockdown. You can have some function and some cells still having that particular unedited version of the gene. And in this case, it's again looking at pigmentation. You can see the stark differences between them. So that's the direction I think some of this work is going to continue to go. And it's pretty exciting to utilize some of these new tools for salmon and fish health. So I'll stop right there. I wanted to make sure I leave some time for questions. Hopefully that was a good high-level overview of kind of the directions of research that our lab is taking, and how it's influenced by issues that occur within industry, and trying to provide sort of applied research for important questions that impact both industry, but even wild fish currently. So there's many collaborators and funders to thank. I mentioned a few. I'll just kind of show a picture of several of them so that I can avoid forgetting many people and providers. So at this time, I'll end, and I'll take any questions you might have. Thank you.
Spectacular presentation, Mark. Thank you so much for the overview. It's fascinating stuff. Genetic approaches to resistance and health are just fascinating. Folks, if you've got questions, feel free to unmute yourself and ask Mark directly, or you can drop questions into the chat. And I can either read them, or Mark, you can keep an eye there too.
Just trying to see if there's any questions in the chat.
I don't see any come in yet. I'll get us started with one, though. I'm curious, for the genetic modification work that you're doing, what sort of biosecurity requirements does Canada require, your university require you to have in place to prevent escape? Or
Yeah.
yeah.
So currently that work is being done at a level 3, aquatic level 3 facility. However, I don't, part of that is we are doing some of that work on non-endemic species to Atlantic Canada. So it was initiated at a level 3 specifically to do that. In terms of doing CRISPR work on an endemic species, so Atlantic salmon here, I'm not sure if we can do that at a level 2 facility or not. That's a great question. We haven't had the interest to do that here yet, because we've kind of already been doing the work at a level 3 facility. But it is something that we've talked about doing. So we do need to know whether we could actually do it here at AVC, where our controls are level 2, or whether we do need to do it at a level 3. So at this point, I don't know. I don't know the answer to that. My expectation is that it's level 2, because I know that there's other labs that are doing CRISPR work, and I don't think that they're level 3 facilities. So that's a guess. I guess I'll have to get back to you on that one, and we do need to figure it out ourselves.
And I'm curious too, a follow-up question on the modification strategies. What's your role as a researcher, technology developer, and sort of the regulatory and ethical conversations of applying this approach for production fish?
Um, so on the editing side.
Yeah. So yeah, what are you involved in those conversations on how this might move through a regulatory approval process, for example?
No, so as part of our work with NoFima and collaborators in Norway, there's actually a group within NoFima that's having some of those regulatory discussions. Again, that's with the Norwegian regulatory bodies. I have not had any of that conversation here in Canada, so that would be, I guess, above my pay grade. But it's a great question. Now on the ethical side too, one of the what's some really interesting modeling work that's being done at University of Melbourne by a couple of our collaborators, looking at how a gene edit might pass through a population of wild fish, with a point source being, in this case, it would probably be a farm, right? And how it might move through an industry or a wild population. And so there's been a couple papers that have been published just in the last year or two, looking at that spread and using Norway as an example. I'm trying to think. It's who is it again? Coates is the lead author, and he's one of the guys that's collaborating on this huge project. And if anybody has interest on some of that, I could always forward you the paper or the PDF, because I know that they've published one or two papers on that idea. But it is an area that there is a group kind of seriously considering what are the ramifications of releasing, I guess you could say, an edit into the field. What would it do? How would it spread? Because you're right that these are very important questions we need to answer before we even consider something like that. Right now, we use it as a tool to kind of better understand how what are the controls for sensitivity and resistance, but the logical step at some point might be to go out and use it in terms of a farm application. So we do need to have a better understanding of what the impacts are.
Very cool. We had a couple of questions here in the chat, Mark.
Okay.
Dr. Salinas is interested in a PhD student position if you're accepting them.
Yeah, right.
She's
I
got
should do one with her.
she's got a great question here. Do you think that repeated damage by jellyfish impacts stem cell memory at the epigenetic level in the gills and their ability to regenerate?
Wow, that's a great question. I think that's a whole other question, right? We've looked at repeated damage, not from jellyfish, but more from the other treatments, right? Like, so using hydrogen peroxide, but that's been very kind of moderate in terms of the impact. It's acute, it's transient, it's with hours to maybe a few days. The impact we tried with our jellyfish work to kind of go almost kind of subclinical or not be as impactful as there's been a couple other groups that have looked at jellyfish impacts and mortality caused due to jellyfish. So we kind of went to look at kind of the low grade or low level impacts on the gill in terms of trying to maintain welfare in those animals. So it's a great question. I don't have a good answer. I think in terms of the epigenetic impacts, that's an area that we really need to get a better handle on in salmon in general, I think, or fish in general, because there are so many epigenetic controls that we're just learning are important in day-to-day responses immunologically, physiologically in these fish. I think the regeneration work, that model is a great setup for testing just that. Not necessarily the jellyfish impact, but stem cell memory in gills that have gone through the repeated resection. So I think that's a great question, Irene, and I think we could begin to answer that if we continue on that regeneration avenue with Faru. She's an excellent postdoc. She's probably going to get a job in the next week or two or something because she's doing such excellent work. But I would really like to see further development of that regeneration work, because I think it as we got into the gill health work, we began to realize, I think, that we don't have a lot of answers just in terms of the ability of these gills to regenerate in salmon. There's been some work in zebrafish and a few other species. So we actually, Faru went back and kind of started from very basic examination of the ability and how much can be removed before the filaments don't regenerate. So there's a lot of work, I think, there that needs to be done.
Thank you. I love it. It's so good.
It's great to see you.
You too.
So I'd say definitely, Irene, check out Faru's work. I just gave one slide on it, but she's got a couple publications on it that have just come out. And she's doing some really cool RNA scope of some of these markers of regeneration right now. And I think she'll be presenting that over the next probably 6 months too. So keep an eye out for that.
Hi, Mark. Can you hear me?
Yep.
This is Jessie, University of Idaho. And
Yep.
I have a question because for your CRISPR-Cas9, the plasmid you used, did you edit it before you used it, or you just use the commercial plasmid directly? Because I noticed I did the CRISPR-Cas9 when I was a PhD student, that's 10 years ago. And I did it on the EPT cells and also zebrafish. And I
Yep.
noticed the commercial ones, maybe I used the first generation CRISPR-Cas9 plasmid, but at that time, the promoters, the MIMO promoters, and then they didn't show really good function, and they didn't show good FC when you work on the either the EPT cells or the zebrafish. So that's why I'm wondering if you have the updated one with the fish promoter and then show really good function, or still like the same like MIMO promoters.
Yeah. So, I mean, I think there's different promoters that you can use for sure. We've used for the CRISPR-Cas9 that we looked at. We purchased the enzyme commercially, and it would be, I guess, the most recent version. But we had there's collaborators in Norway that actually did the individual target spacers. So how they because that generated the 3 different targets per knockout that we wanted to look at. If you look at Edvardson's paper, so there's a Rolf Edvardson, he heads up a group in Norway, and I forget the name of the institute. It's Norwegian, so I always bungle it. If Lars is still here, he could probably say it properly. But they did a 2014 paper in salmon looking at knocking out 2 different pigment genes. We actually followed that protocol almost to a T, except what we changed was, and if you want to email me after, I can send you the paper. What we changed was we actually looked at multiple targets for the same gene within the same embryo. So we actually knocked out 3 to 4 targets in an individual embryo to knock out a particular gene with function that we were interested in. So there's like the design of the targets too might be different than the design from 10 years ago in terms of the actual spacer and where it connects. I'm not familiar with kind of the older technology, but definitely reach out to me. My email is mfast@upi.ca, and I can share the Edvardson's paper and our methodology for you if you like.
Thank you, Mark. I will definitely email you later.
No problem.
Yeah, thank you.
Folks, if you have take questions, folks want to unmute themselves first. We're running out of time. We've got a few in the chat though.
Look at Lars. Great job, Lars. So it's in the chat group there, in the chat function there. Rolf Edvardson, Institute of Marine Research in Bergen. I thought there was a Norwegian name for it. Maybe that's what I'm getting mixed up. I could have said Institute of Marine Research. Geez, that's pathetic. Thanks, Lars.
So from the chat, Mark, for the mRNA vaccines, what age fish are you vaccinating? And do you see long-term protective immunity?
Yeah, great question. So we've just started in that area, and I didn't talk about it today. We're vaccinating 20-gram fish is what we're starting with. However, we've done boosts of the vaccines just prior to seawater entry. So those are at about 90 to 100 grams, but we haven't had any success in the boost showing any impact. So that could be that, well, the boost just doesn't work. It could also be that we're not giving enough at the boost because now the fish is 5 times larger. So there are some things on the boosting that we need to work out. In the mRNA vaccine that we're currently working on is against infectious salmon anemia virus. So it's a saltwater pathogen. So we want to vaccinate the fish around the size that they're generally going to be vaccinated in the industry. So that's why we chose 20 grams. But as you can imagine, if they're growing to 100 grams and maybe even now with industry moving to larger smolts, they might be 200 grams by the time they go to sea. So vaccinating at 20 grams, we need a much longer protective window to be able to have an impact once those fish get out to sea. Right now, though, we're just working, as I said, the smaller sizes. We're seeing protection when exposed. I think the longest we had was it would have been maybe a month after seawater entry. So these fish were, 150, 200 grams approximately, and were exposed then and done in the lab. These weren't field exposures. But we haven't seen protection when done 8 to 12 months later. Like I'd have to double check the degree days, but we did do a study where we looked at our top 4 candidates and let the fish stay in seawater for an extended period and then challenge them later. And we didn't see the same protection. So getting back, we aren't seeing the kind of protection that would be required to protect a fish for the 19 months out at sea. That being said, I think you could probably look, you'd have a hard time finding vaccines that are out there that have a lot of data around them that do protect that long. That's, I think, one of our poorest areas of understanding is length of protection of these vaccines that are out there in terms of their ability to protect for, say, 2 years or 3 years after administration. And that's just because it's not easy to do, but it's an important question that needs to be answered. So thanks for asking that.
That's great, Mark. We're going to end it there for the sake of time. Your email's in the chat, though, if folks want to follow up with you directly. I'm sure you're cool with that.
Yeah,
lots of compliments coming in, too. Thanks again. Excellent presentation. Very interesting stuff, as always.
Thank you very much for the opportunity. It's great to see everybody, and always great. I know my students and I enjoy this seminar series. So really, thanks for putting it on, guys. It's been great.